Hello students, in today's discussion we will cover chapter 9 lesson 2, the process of cellular respiration. Now let's see the learning objectives. Describe what happens during glycolysis. Describe what happens during the Krebs cycle. Explain how high energy electrons are used by the electron transport chain. And the last one is to identify how much ATP cellular respiration generates. Well, students, for your information, you can find this lesson on your textbook pages 254 to 257. Now, let's go to the first stage of cellular respiration, that's the glycolysis. So in our discussion, we will try to answer what happens during the process of glycolysis. Generally, what happens during glycolysis is one molecule of glucose, that's a 6-carbon compound, is transformed into 2 molecules of pyruvic acid, which is a 3-carbon compound. Now let's see the detail. So glycolysis is the first stage of cellular respiration, as you know. So during glycolysis, glucose is broken down into 2 molecules of the 3-carbon molecule, pyruvic acid. So pyruvic acid is a reactant in the Krebs cycle. That means it will be used to run the Krebs cycle. And ATP and NADH, ATP and NADH are produced as part of the process as well. Well, now let's see the ATP production. So the cell deposits 2 ATP molecules. It deposits 2 ATP molecules into its account to get glycolysis going. And glycolysis then produces 4 ATP molecules, giving the cell a net gain of 2 ATP molecules for each molecule of glucose that enters glycolysis. Well, now let's see the NADH production. NADH carries the high energy electrons to the electron transport chain, where they can be used to produce more ATP. So 2 NADH molecules are produced for every molecule of glucose that enters the glycolysis. Okay, now the advantage of glycolysis. Glycolysis produces ATP very fast, which is an advantage when the energy demands of the cell suddenly increase. Another advantage is that glycolysis does not require oxygen, so it can quickly supply energy to cells when oxygen is unavailable. Now let's jump to the Krebs cycle. So in our discussion, we will see what happens during the Krebs cycle. Generally speaking, during the Krebs cycle, pyruvic acid is broken down into carbon dioxide in a series of energy extracting reactions. Well, now let's see the Krebs cycle, that is the second stage of cellular respiration. During the Krebs cycle, the second stage of cellular respiration, pyruvic acid produced in glycolysis is broken down into carbon dioxide. It's broken down into carbon dioxide in a series of energy extracting reactions. The Krebs cycle is also known as the citric acid cycle because citric acid is the first compound formed in the series of reactions. Now let's see the citric acid production. So pyruvic acid from glycolysis enters the matrix of the innermost compartment of the mitochondrion, and once pyruvic acid is in the mitochondrial matrix, the NAD accepts two high energy electrons to form NADH, and one molecule of carbon dioxide is also produced. The remaining two carbon atoms react to form acetyl-CoA. So acetyl-CoA combines with the acetyl-CoA combines with four carbon molecules to produce citric acid. Energy released by the breaking and rearranging of carbon compounds is captured in the forms of ATP, NADH, and FADH. So for each turn of the cycle, one ADP molecule is converted into ATP. ATP can directly power the cell's activity. The electron carriers NAD and FAD each accept pairs of high energy electrons to form NADH and FADH2. NADH and FADH are used in the electron transport. They are used in the electron transport chain to generate ATP. So each molecule of glucose results in two molecules of pyruvic acid, which enter the Krebs cycle. So each molecule of glucose results in two complete turns of the Krebs cycle. Therefore, for each glucose molecule, 6 carbon dioxide molecules and 2 ATP molecules, 8 NADH molecules, and 2 FADH molecules are produced. Well, now let's see the third stage of cellular respiration. That's the electron transport chain. So in our discussion, we will see how does the electron transport chain use high energy electrons from glycolysis and the Krebs cycle. Generally, the electron transport chain uses the high energy electrons from glycolysis and the Krebs cycle to convert ADP into ATP. Now let's see the process in detail. NADH and FADH pass their high energy electrons to electron carrier proteins in the electron transport chain. So at the end of the electron transport chain, the electrons combine with hydrogen ions and oxygen to form water. So the hydrogen ions and oxygen combine to form water. So energy generated by the electron transport chain is used to move hydrogen ions against a concentration gradient across the inner mitochondrial membrane and into the intermembrane space. Hydrogen ions pass across the mitochondrial membrane through the ATP synthase, causing the ATP synthase a molecule to spin. With each rotation, the ATP synthase attaches a phosphate to ADP to produce ATP. Now let's see the totals. So how much energy does cellular respiration generate? Together, glycolysis, the Krebs cycle, and the electron transport chain release about 36 molecules of ATP per molecule of glucose. Let's see the energy totals. So in the presence of oxygen, the complete breakdown of glucose through cellular respiration results in the production of 36 ATP molecules. This represents about 36% of the total energy of glucose. The remaining 64% is released as heat. Generally, the cell can generate ATP from just about any source. Even though we have modeled it using only glucose, in our discussion, complex carbohydrates are broken down into simple sugars like glucose. Also, lipids and proteins can be broken down into molecules that enter the Krebs cycle or glycolysis at one of several places. Students, this is all about our discussion. For more information, you can watch the videos that are represented in your weekly plan by clicking the links given there. Now, now let us go to the section assessment questions. Question 1A. What are the products of glycolysis? Here is the answer. 2 molecules of pyruvic acid, 2 molecules of NADH, and a net gain of 2 ATP molecules. Let's see 1B. How is the function of NAD similar to that of NADP? So both molecules are electron carriers. Both molecules are electron carriers. So let's see 2A. What happens to pyruvic acid in the Krebs cycle? What happens is just pyruvic acid is broken down into carbon dioxide through a series of energy extracting reactions collectively called the Krebs cycle. Let's see 2B. Okay, just look at figure 9.5 and list the products of the Krebs cycle. What happens to each of these products? You have to refer figure 9.5. So the answer is the products are ATP, NADH, FADH2, and CO2. CO2 is expelled in exhalation. The ATP molecules are used to power cellular activities. The NADH and FADH2 molecules are used in the electron transport chain to generate ATP. Let's see 3A. How does the electron transport chain use the high energy electrons from glycolysis and the Krebs cycle? The answer is to power the pumping of hydrogen ions against a concentration gradient from the matrix to the intermembrane space. Let's see 3B. How does the cell use the charge differences that build up across the inner mitochondrial membrane during cellular respiration? Here is the answer. The charge differences force protons through ATP synthase, which powers the conversion of ADP to ATP. Now let's see the last question. For A, how many molecules of ATP are produced in the entire breakdown of glucose. How many molecules of ATP are produced? About 36 molecules of ATP per molecule of glucose. Well, now for B, how is the cell like a furnace? So it can burn many different types of fuels, not just glucose. Also, a cell releases heat energy through the breakdown of glucose that heats the organism, just as a furnace releases energy to heat a building.